Components of the blood. Hemoglobin is responsible for distributing oxygen, and to a lesser extent, carbon dioxide, throughout the circulatory systems of humans, vertebrates, and many invertebrates. The blood is more than the proteins. Though, blood is actually a term used to describe the liquid that moves through the vessels and includes plasma, the liquid portion, which contains water, proteins, salts, lipids, and glucose, and the cells, red and white cells, and cell fragments called platelets. Blood plasma is actually the dominant component of blood and contains the water, proteins, electrolytes, lipids, and glucose. The cells are responsible for carrying the gases, red cells, and immune the response, white. The platelets are responsible for blood clotting. Interstitial fluid that surrounds cells is separate from the blood, but in hemolymph, they are combined. In humans, cellular components make up approximately 45% of the blood and the liquid plasma 55%. Blood is 20% of a person's extracellular fluid and 8% of weight. The role of blood in the body. Blood, like the human blood illustrated in Figure 1, is important for regulation of the body's systems and homeostasis. Blood helps maintain homeostasis by stabilizing pH, temperature, osmotic pressure, and by eliminating excess heat. Blood supports growth by distributing nutrients and hormones, and by removing waste. Blood plays a protective role by transporting clotting factors and platelets to prevent blood loss, and transporting the disease-fighting agents or white blood cells to sites of infection. Red blood cells, or erythrocytes. Erythra equals red, cyte equals cell, are specialized cells that circulate through the body delivering oxygen to cells. They are formed from stem cells in the bone marrow. In mammals, red blood cells are small biconcave cells that at maturity do not contain a nucleus or mitochondria, and are only 7 to 8 micrometers in size. In birds and non-avian reptiles, a nucleus is still maintained in red blood cells. The red coloring of blood comes from the iron-containing protein hemoglobin, illustrated in Figure 2. The principal job of this protein is to carry oxygen, but it also transports carbon dioxide as well. Hemoglobin is packed into red blood cells at a rate of about 250 million molecules of hemoglobin per cell. Each hemoglobin molecule binds 4 oxygen molecules so that each red blood cell carries 1 billion molecules of oxygen. There are approximately 25 trillion red blood cells in the 5 liters of blood in the human body, which could carry up to 25 sextillion, 25 times 10^21, molecules of oxygen in the body at any time. In mammals, the lack of organelles in erythrocytes leaves more room for the hemoglobin molecules, and the lack of mitochondria also prevents use of the oxygen for metabolic respiration. Only mammals have enucleated red blood cells, and some mammals, camels, for instance, even have nucleated red blood cells. The advantage of nucleated red blood cells is that these cells can undergo mitosis. Enucleated red blood cells metabolize anaerobically, without oxygen, making use of a primitive metabolic pathway to produce ATP and increase the efficiency of oxygen transport. Not all organisms use hemoglobin as the method of oxygen transport. Invertebrates that utilize hemolymph rather than blood use different pigments to bind to the oxygen. These pigments use copper or iron to the oxygen. Invertebrates have a variety of other respiratory pigments. Hemocyanin, a blue-green, copper-containing protein, illustrated in Figure 2b, is found in mollusks, crustaceans, and some of the arthropods. Chlorochorin, a green-colored, iron-containing pigment, is found in 4 families of polychaete tubeworms. Hemerythrin, a red, Iron-containing protein is found in some polychaete worms and annelids and is illustrated in Figure 2c. Despite the name, hemerythrin does not contain a heme group and its oxygen-carrying capacity is poor compared to hemoglobin. The small size and large surface area of red blood cells allows for rapid diffusion of oxygen and carbon dioxide across the plasma membrane. In the lungs, carbon dioxide is released and oxygen is taken in by the blood. In the tissues, oxygen is released from the blood and carbon dioxide is bound for transport back to the lungs. Studies have found that hemoglobin also binds nitrous oxide. NO is a vasodilator that relaxes the blood vessels and capillaries and may help with gas exchange and the passage of red blood cells through narrow vessels. Nitroglycerin, a heart medication for angina and heart attacks, is converted to NO to help relax the blood vessels and increase oxygen flow through the body. A characteristic of red blood cells is their glycolipid and glycoprotein coating. These are lipids and proteins that have carbohydrate molecules attached. In humans, the surface glycoproteins and glycolipids on red blood cells vary between individuals, producing the different blood types, such as A, B, and O. Red blood cells have an average lifespan of 120 days, at which time they are broken down and recycled in the liver and spleen by phagocytic macrophages. A type of white blood cell. White blood cells. White blood cells, also called leukocytes, leuko equals white, make up approximately 1% by volume of the cells in blood. The role of white blood cells is very different than that of red blood cells. They are primarily involved in the immune response to identify and target pathogens, such as invading bacteria, viruses, and other foreign organisms. White blood cells are formed continually. Some only live for hours or days, but some live for years. The morphology of white blood cells differs significantly from red blood cells. They have nuclei and do not contain hemoglobin. The different types of white blood cells are identified by their microscopic appearance after histologic staining, and each has a different specialized function. The two main groups, both illustrated in Figure 3, are the granulocytes, which include the neutrophils, eosinophils, and basophils, and the agranulocytes, which include the monocytes and lymphocytes. Granulocytes contain granules in their cytoplasm. The agranulocytes are so named because of the lack of granules in their cytoplasm. Some leukocytes become macrophages that either stay at the same site or move through the blood stream and gather at sites of infection or inflammation where they are attracted by chemical signals from foreign particles and damaged cells. Lymphocytes are the primary cells of the immune system and include B cells, T cells, and natural killer cells. B cells destroy bacteria and inactivate their toxins. They also produce antibodies. T cells attack viruses, fungi, some bacteria, transplanted cells, and cancer cells. T cells attack viruses by releasing toxins that kill the viruses. Natural killer cells attack a variety of infectious microbes and certain tumor cells. One reason that HIV poses significant management challenges is because the virus directly targets T cells by gaining entry through a receptor. Once inside the cell, HIV then multiplies using the T cell's own genetic machinery. After the HIV virus replicates, it is transmitted directly from the infected T cell to macrophages. The presence of HIV can remain unrecognized for an extensive period of time before full disease symptoms develop. Platelets and coagulation factors. Blood must clot to heal wounds and prevent excess blood loss. Small cell fragments called platelets, thrombocytes, are attracted to the wound site where they adhere by extending many projections and releasing their contents. These contents activate other platelets and also interact with other coagulation factors, which convert fibrinogen, a water-soluble protein present in blood serum, into fibrin, a non-water-soluble protein, causing the blood to clot. Many of the clotting factors require vitamin K to work, and vitamin K deficiency can lead to problems with blood clotting. Many platelets converge and stick together at the wound site, forming a platelet plug, also called a fibrin clot, as illustrated in Figure 4b. The plug or clot lasts for a number of days and stops the loss of blood. Platelets are formed from the disintegration of larger cells called megakaryocytes, like that shown in Figure 4a. For each megakaryocyte, 2,000 to 3,000 platelets are formed, with 150,000 to 400,000 platelets present in each cubic millimeter of blood. Each platelet is disc-shaped and 2 to 4 micrometers in diameter. They contain many small vesicles but do not contain a nucleus. Plasma and serum. The liquid component of blood is called plasma, and it is separated by spinning or centrifuging the blood at high rotations, 3,000 revolutions per minute or higher. The blood cells and platelets are separated by centrifugal forces to the bottom of a specimen tube. The upper liquid layer, the plasma, consists of 90% water along with various substances required for maintaining the body's pH, osmotic load, and for protecting the body. The plasma also contains the coagulation factors and antibodies. The plasma component of blood without the coagulation factors is called the serum. Serum is similar to interstitial fluid, in which the correct composition of key ions acting as electrolytes is essential for normal functioning of muscles and nerves. Other components in the serum include proteins that assist with maintaining pH and osmotic balance, while giving viscosity to the blood. The serum also contains antibodies, specialized proteins that are important for defense against viruses and bacteria. Lipids, including cholesterol, are also transported in the serum, along with various other substances including nutrients, hormones, metabolic waste, plus external substances, such as drugs, viruses, and bacteria. Human serum albumin is the most abundant protein in human blood plasma and is synthesized in the liver. Albumin, which constitutes about half of the blood serum protein, transports hormones and fatty acids, buffers pH, and maintains osmotic pressures. Immunoglobin is a protein antibody produced in the mucosal lining and plays an important role in antibody-mediated immunity. Evolution connection. Blood types related to proteins on the surface of the red blood cells. Red blood cells are coated in antigens made of glycolipids and glycoproteins. The composition of these molecules is determined by genetics, which have evolved over time. In humans, the different surface antigens are grouped into 24 different blood groups with more than 100 different antigens on each red blood cell. The two most well-known blood groups are the ABO, shown in Figure 5, and Rh systems. The surface antigens in the ABO blood group are glycolipids, called antigen A and antigen B. People with blood type A have antigen A. Those with blood type B have antigen B. Those with blood type AB have both antigens. And people with blood type O have neither antigen. Antibodies called agglutinogens are found in the blood plasma and react with the A or B antigens if the two are mixed. When type A and type B blood are combined, agglutination, clumping, of the blood occurs because of antibodies in the plasma that bind with the opposing antigen. This causes clots that coagulate in the kidney, causing kidney failure. Type O blood has neither A or B antigens, and therefore, type O blood can be given to all blood types. Type O negative blood is the universal donor. Type AB positive blood is the universal acceptor because it has both A and B antigen. The ABO blood groups were discovered in 1900 and 1901 by Karl Landsteiner at the University of Vienna. The Rh blood group was first discovered in rhesus monkeys. Most people have the Rh antigen, Rh+, and do not have anti-Rh antibodies in their blood. The few people who do not have the Rh antigen and are Rh can develop anti-Rh antibodies if exposed to Rh plus blood. This can happen after a blood transfusion or after an Rh woman has an Rh plus baby. The first exposure does not usually cause a reaction. However, at the second exposure, enough antibodies have built up in the blood to produce a reaction that causes agglutination and breakdown of red blood cells. An injection can prevent this reaction. Section summary. Specific components of the blood include red blood cells, white blood cells, platelets, and the plasma, which contains coagulation factors and serum. Blood is important for regulation of the body's pH, temperature, osmotic pressure, the circulation of nutrients and removal of waste, the distribution of hormones from endocrine glands, and the elimination of excess heat. It also contains components for blood clotting. Red blood cells are specialized cells that contain hemoglobin and circulate through the body, delivering oxygen to cells. White blood cells are involved in the immune response to identify and target invading bacteria, viruses, and other foreign organisms. They also recycle waste components, such as old red blood cells. Platelets and blood clotting factors cause the change of the soluble protein fibrinogen to the insoluble protein fibrin at a wound site, forming a plug. Plasma consists of 90% water along with various substances, such as coagulation factors and antibodies. The serum is the plasma component of the blood without the coagulation factors. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.